Welcome back, Biology 104. Today we're going to be talking about diversity, and this covers pages 7 through 12 in your notes. So, the topic that these different concepts we're going to look at would be under what's called systematics, and that is the study of the kinds and diversity of organisms and the evolutionary relationships among them. So, it's looking at how are these organisms, how can they be classified? How are they related, historically and evolutionarily speaking? So within systematics, you'd be looking at taxonomy, classification, and binomial nomenclature. So what is their genus, species, name? How do we write that? And then what other groups or taxons or organisms going to be within? And then also with systematics would be looking at that evolutionary history. So a timeline is included in systematics. So for taxonomy specifically, that's going to be looking at the original description of the species. What work is involved in describing that species, and then how do we group it, or how is it classified? So this would have come before systematics, actually. Before there was really a developed any kind of evolutionary hypothesis. Before that, Linnaeus was looking at, well, how do we even group all these animals that we have and all these organisms and all kinds of life forms, all life? How do we organize all of it first? And then there was the theories of evolution. How did they arise? So classification is the ordering of these living things into what we call a taxon, or you can think of these as large groups, and it's a hierarchical system. So what that means is the higher taxons are going to include more organisms, but every time you go up a taxon, you actually have less groups within that taxon. So for example, there's only, if you get down to, well, let me give you the list here. So the list of taxons from top to bottom, and this looks like a big upside down triangle with larger squares on the top representing the first term, and then smaller squares underneath as you get to the lower taxons. So at the very top of the taxons, you have domain, then kingdom, then phylum, class, order, family, genus, species. You have to know all of those taxons, you have to know all their names, and you also have to know their order, and which one is most specific and which one is least specific. So I'll go through that with you now. So one way to remember the order is this mnemonic device. Dominating King Philip came over for good soup. If you can remember that, that gives you the order. Dominating, domain, King Philip, kingdom, phylum, came over for class, order, family, and then good soup is genus, species. So that gives you the order, and just know that you start at the least specific. The domain is the least specific taxon to the most specific. On the bottom of that would be species. So that's at the very bottom of that upside down triangle of stacked squares or rectangles. And basically what it is a way to classify all the living things on the planet. So for example, if we look at kingdom, right, the animal kingdom, so it goes domain, kingdom. Well, within the animal kingdom, all of those organisms are going to have some shared characteristics. They're all going to be made of animal cells, for example. So everything that an animal cell has will be shared between everything as you move past and underneath the animal kingdom to specific phylum, classes, orders, families, etc. But if you get to like two different kingdoms, they're going to have different traits. So anyway, we'll go through what those specific kingdoms are here in a minute, and you'll see in more detail as we kind of flesh this out. But Linnaeus was the one to develop a nomenclature system and our classification system that we just looked at. So binomial nomenclature is the system that gives each species a two-word name. So the binomial nomenclature and classification system was created by him, and I'll show you how to write those here in just a little bit. But it's basically when you mention an animal or an organism, you always give its genus and species name. That's its two-word names, and that's why it's called binomial, two name, and then nomenclature would be the system of naming that we have. And Carl Linnaeus was an interesting character, actually. He was extremely intelligent, obviously. And when he developed his system of binomial nomenclature, he became extremely renowned for that. And he was actually what they call ennobled in 1761. And he became royalty at that point. He became part of the nobility. And he did that for his ordering of the natural world, which was a really popular task when it was achieved by him. And he's become known as the father of taxonomy, which is pretty neat. But anyway, that's why his name changed from Carl Linnaeus to Carl von Linné. So again, you need to learn these 8 taxons and their order. And if you just remember, dominating King Philip came over for good soup, and that's in order from least specific to most specific, then you can remember the 8 taxons. So for that, the only ones that you're going to have to memorize is going to be the domains and the kingdoms. Everything else, you'll actually probably memorize some of them just because they'll come up more often than others. But these are the ones that you're going to have to memorize. So everybody should know the 3 domains of life. And the 3 domains are bacteria, archaea, and eukarya. Every living thing on the planet fits into one of these 3 categories. And just to give you a brief description of what these categories are like, these domains, of course, bacteria, these are extremely simple organisms in comparison to other more highly developed organisms. But bacteria are generally really simple, really small. They're unicellular, and they're prokaryotes, which means they do not have a nucleus in their cells. Another type of organism, which seems very similar, would be the archaea. And they're usually described as like super bacteria or extreme bacteria. And they're very ancient organisms with a very ancient origin. They're also unicellular, meaning made of one cell. And they're also prokaryotes, which means they do not share or they do not have a nucleus. In the diagram, you'll see this family or tree of life here branching out. And actually, archaea are more closely related to eukarya than are bacteria, although they very much resemble bacteria. And in fact, we describe them as superbacteria. But anyway, eukarya would be the third group, and that's what we call the eukaryotes. So our examples of those would be protists, fungi, animals, and plants. They're going to be more complex. They can be, and often are, multicellular. They're going to have a nucleus and organelles in their cells and things of that sort. So that would be the 3 domains. Bacteria, archaea, eukarya. The 6 kingdoms, these are the 6 kingdoms. 2 of them you can remember because they're just based on the domain names that they come from. So for example, archaea is one of the 6 kingdoms. It's called Archaebacteria. And then the other, or the second of the 6 kingdoms, is eubacteria, which is just from the domain bacteria. So 2 of the 6 are just 2 of the domains, Archaebacteria and eubacteria. The other 4 are 4 different kingdoms under the domain eukarya. And the 4 kingdoms of eukaryotes are Protista, fungi, plantae, and animalia. Protist, fungus, plants, and animals. Those are your 4 kingdoms within the eukaryotic domain. So when we look at those 4 kingdoms, well, they're all eukaryotes. So each one of those cell types is going to have a nucleus, and they're going to have organelles and things of that sort. So they're going to share traits with each other because they're all under the same domain. But because they're in different kingdoms, they're going to have different characteristics. So you can see the usefulness of this organizational system using these taxons and this classification. So originally, when I was working on this set of notes, I really wanted a chart like this, and I couldn't find one anywhere. And basically what this chart is it lists how many of each taxon there are within each taxon, how many groups within it. how many kinds. So for example, domain, there are 3, kingdoms, there are 6. And when I started doing research on the others to get a clear-cut number, I realized that, the numbers of phyla, for example, are not settled. There are different debates about how many phyla there should be, how many classes there should be. But I used AI, of course, to try to summarize what's a range of numbers within these different taxons. And you can kind of see the trend. So for example, there's 3 domains: archaea, eukarya, and bacteria. And then there's 6 kingdoms: archaebacteria, eubacteria, protista, fungi, plantae, and animalia. But then you get to phylum, and there are 53 approximately phyla, depending on how it's organized. So you can see it increases 3, 6, 53. Then to classes, you get up to 200 classes. Orders, 700. Family, 2,800. Genus, 38,000. And then species, we have scientists have described 2 million different types of species. And they think that that number could end up being, eventually, anywhere from 10 million to 1 trillion different types of species. So you can see that there's more organisms as you move down, because there's more with groups within each taxon as you go down. And that's because you start to notice more differences, and those differences need to be divided into different groups. And because species means one specific thing, there's going to be millions of those, because there's millions of different types of living things on the planet, which is kind of amazing. It's kind of hard to even imagine. But there are, especially when you look at bacteria and really tiny organisms like insects. There's a great variety there. So, taxons can also be placed into larger groups or subdivided into smaller groups. And we use certain prefixes when this happens. You might see super or sub or infra as a prefix to describe these different groupings. So for example, the phylum Chordata would be the chordates, and you could have underneath that the subphylum Vertebrata or the vertebrates. So just know that can also be used. Here's an example of that taxon classification. So for example, for the gray wolf, you would have Eukarya for the domain, Animalia for the kingdom, Chordata for the phylum, Mammalia for the class, Carnivora for the order, Canidae for the family, Canis for the genus, and then the species could be Lupus, for example, the gray wolf, Canis lupus, or for the coyote, it could be Canis latrans. So you'll notice they share the genus name, but then the species name is specific to that one type of species. And then here's another example that I won't go through, but just with the brown bear, the Ursus arctos. And you'll notice, right, domain, there's lots of organisms, but then as you move down, it gets more and more narrow and narrow until you get to one specific species. And the way we describe the species and how Carl Linnaeus did is they use what's called binomial nomenclature, which is a 2-word name. The first name is the genus, the second name is the species, or what they call the specific epithet. So together, the genus and the species is what we describe as the scientific name. So if you're reading a scientific paper and you see something like Squalus acanthius, and it's written italicized, the first word is capitalized, second word is lowercase, that is from that binomial nomenclature of Carl Linnaeus. And it usually has some kind of like a Latin name to it, because that's what they've used for a long time in science. And that's carried over, and they, even if they name something new, they'll try to give it a Latin sounding name or use a Latin word. But the genus name is capitalized. Both names are either underlined or italicized, and the species name is lowercase. So in the name Squalus acanthius, the S in Squalus is capitalized, the A in acanthius is lowercase, and it's all italicized. And whoever names the organism is called its authority. And Carl Linnaeus is the one who named actually the dogfish, which is a shark, and he gave it that name, Squalus acanthius. And so many of their names are usually like some type of Latin word or Greek word. in its root that means something about the way it looks oftentimes. So, for example, this one, Melanerpes erythrocephalus, is the genus species name, and this is a red-headed woodpecker. Well, Melanerpes, you think about melanin, color, erythrocephalus. Erythro would be red, that's what it means, like erythrocytes, which are red blood cells. And then cephalus means head, so it means melanin of red head, basically, or a red-headed organism. And this organism has a red head. And so anyway, the names will often correlate to their appearances. So that brings us to cladistics. So cladistics is where we have these clades, or these diagrams where we branch off to show how organisms are related. And for this, we create what's called a cladogram, which are those charts where you have a line, and it's like a timeline, and then other lines branch off of them to describe how those organisms are related to each other. You could think of it like a family tree, right? You have the grandparents, and then you draw a line down from the grandparents to their kids, and then lines from their kids and their kids' spouses to the grandkids, and so on and so forth. It's sort of like that. Now, the premise or the assumption for these charts is common ancestry. You're assuming that they share a related history, or that they share a common ancestor when you draw these diagrams. So if you draw two organisms, two clades that are close to each other on the end of your cladogram, that means you're demonstrating or hypothesizing that they are more closely related. And those that are on opposite ends of a cladogram, you would predict to be more distantly related. You will show shared traits and derived traits, and we'll show you what that is as we go. Now, there is a difference between a cladogram and a phylogram, or a phylogenetic tree. And the difference is a cladogram will show how organisms branch off and how they're related, which ones share more recent or more distant common ancestors. But the lengths of those lines don't correlate to like an actual time period, not in any exact sense. Whereas a phylogram or a phylogenetic tree tries to represent the length of the branches to correlate to the evolutionary time, or the time that has passed. So for those, the lengths of the lines actually do matter. Whereas in a cladogram, it doesn't matter. It's just showing the relatedness without accounting for time. So here's an example of a cladogram. So if you can imagine a line going from the bottom left to the top right, just a diagonal line. And then about halfway up that line, you have a branch that comes off. So it looks like the letter Y, basically, is what this cladogram looks like. And at the base of that Y, on the very bottom on the left, you have the letter A, and that represents an organism. As you move up the letter Y, where it branches to the left, at the end of that branch, we have the letter B. And when it branches to the right, at the end of that letter, or at the end of that branch, we have the letter C. So it's A, B, C, from left to right, bottom to top. And each of these letters represents an organism. So organisms A, B, and C all have a vertebral column, is what's being described in this image. Because underneath, along the straight line on that Y, at the base of that, you have traits that are written across that line. And as branches come off, in this case it's like the letter Y, so you only have one branch, those describe, the branches will list organisms, but on the base of a cladogram, where you have a straight line or a diagonal line, it lists traits. So every cladogram will have a list of organisms and a list of traits. And the traits are on the bottom and the organisms come up on the top usually. Sometimes it's written upside down. I'll try to describe those that aren't, even though in this PowerPoint I will have at least one that's upside down. But anyway, so you have these three organisms, A, B, C, and you have these two traits on the bottom. Vertebral column at the base, and then as you move up where it branches, you have lungs. So the question is, well, what would be the ancestral or the primitive character? So what trait pops up first? Well, that's going to be the vertebral column. And then what would be the derived or newly evolved character trait? That would be the lungs, because it comes later in the cladogram, right where that branch occurs in the Y. So which organism would be the ancestor? In that case, that's going to be organism A, because it is at the base of this cladogram. So you can think of A as like the grandparent organism, and then B and C is like the grandkids, and they come out later. And as time passes, organisms could acquire new traits, and they could change over time or adapt or even lose traits, depending on what we're talking about. So cladistics argues that only shared derived characters could possibly give information about phylogeny. Cladistics is intended to be more objective and less subjective than traditional taxonomy, and cladistics was created by Wilhelm Hennig, who was a German entomologist. And entomology is actually a very difficult field of study in terms of cladistics, because insects, it's just crazy trying to classify those from what I've read anyway. So in cladistics, the goal is to place organisms into a monophyletic group, a group that consists of a single ancestor and all of its descendants. We call these groups a clade. And these groups of the derived characteristics are often depicted in tree diagrams called a cladogram. So we'll look at one of these and some of the terms that we'll use. So the root of a cladogram, that is the starting point of the tree. A node would represent an ancestor species. So in this diagram, nodes are represented by blue circles and blue squares. So those are coming off of the cladogram. An internal node would be the last common ancestral population that speciated or split to give rise to two or more new species. Terminal node represents a surviving species of the taxon. The in-group is the species or organisms being analyzed. The out-group, you can think of this as the most distant organism or the outside organism that is used to determine the primitive characters and the derived characters. And we'll look at examples of all of this. So our basic assumptions with cladistics would be 1, any group of organisms is related by descent from a common ancestor. That's the obvious assumption, because if you're drawing these lines, you're assuming they come from one another, or not from one another, but from a common ancestor. And when you do that, you might be wrong. You might draw out, a cladogram, and really every cladogram, to some extent, is a hypothesis. It is a educated guess based on characters and traits and DNA sequencing, whatever evidence you can gather. It is a hypothesis of how they are related. You might be wrong, but the assumption is that they are related. When speciation occurs, the existing species will divide into exactly 2 groups. That's another assumption. That's why we can have all these forks as they branch off into 2 groups. And then the third assumption is that characteristics of organisms change over time. So a key to this is parsimony. And this principle states that the simplest cladogram with the least number of steps is more likely to be accurate, or also would be more easily defended. And they call that Occam's razor. So basically, if your cladogram can look 2 ways, one, it looks super confusing and lines are all over the place, and then another one looks really simple and clean, it's more likely the case that the simpler cladogram is the true one, because generally nature favors, the path of least resistance. And if you have some crazy complicated cladogram where an organism loses and gains eyeballs 30 times, it's very unlikely to occur, so that's probably not correct. So here's an example of one of these cladograms. For this one, it has flat lines where it branches up and then it forks and then it goes up and then each line forks again, and you have a series of all these flatter sort of forking lines. So it could look like that, or it could look like this, where it's one diagonal line from the bottom left to the top right. This is usually what we'll use, and then it branches off all the way up, and every branch leads to an organism. And along the long line, the diagonal line, you have a list of traits. So you'll be asked questions about what's more related, what's more distantly related, what traits do organisms share. Well, any organism past a trait has that trait and all of the traits before it. So for example, if we look at this, we have let's pick an organism. So we'll say birds. So birds up here towards the top right of the cladogram, any trait before it, they're going to have because it passes it as you get there. So from the bottom left to the top right, vertebrae, lungs, amniotic egg, CUV light, all of those you will pass through as you move through the diagram to get to birds. So birds would have all of that. So birds and lizards would also share all those traits because they're right beside each other on the cladogram, and all those traits are before them. So terminal and internal nodes. So if we look at this cladogram to the right, the white circles are internal nodes. So they are on the inside of the lines, the main line. So they are representing common ancestor. So anywhere where you see a fork split out into two species, where those lines meet each other is where there's a hypothesized common ancestor, right? We may not even know what that is. Probably don't. But you have two species, maybe two types of birds that are very similar. It's very likely that they came from a common ancestor. So the hypothesis in the chart is this right here. That's what we're predicting to exist that would have led to these two different species. So we're assuming there's some kind of common ancestor here that would have shared traits, between these two organisms. And maybe they were geographically isolated for a really long time, started to look a little bit different. Maybe their bird songs change, and then they quit breeding, in which case they become different species. So at the top, we have the species in this case, these three gray rectangles, and then the nodes on the bottom are going to be the common ancestors. The node farthest back is going to be the common ancestor for many more organisms. So this one down here would represent the point of common ancestry for species 1, 2, and 3, but this one that's higher up the line would be the common ancestor for species 1 and 2. So that's how you would read that. Then we have something called the in-group and the out-group. So the out-group will be a species or organism that does not contain any of the characters present in the in-group. So the out-group is going to be the older organism, the more primitive, the more ancestral. So one of the things we'll practice in class is you'll be given what's called a character table. And maybe it lists 4 different animals and all the traits that they have. Well, whichever organism has none of those traits, that's going to be your outgroup, and you're going to put it first as the first branch in a cladogram. And then past that, you'll put your first trait, then your next most complex organism, and then your next trait, and then your next most complex organism. And all those other organisms are considered the in group. But the first one, the simplest, the most primitive, that is considered the out group. And we have these certain terms that we use for the characters that we talk about within these charts. So a primitive or ancestral character we call plesiomorphic. And the way you can remember that is plesiomorphic starts with a P, P for primitive. It's going to be the first thing. And then those traits that come after that, we call those derived characters. They have come later, and we call it apomorphic. So here's an example of a cladogram, and we'll go through some questions that you might be asked. So this cladogram goes from sharks. So it's a big, line, diagonal line, bottom left to top right, with tons of branches. And there's traits listed on the main line. And then after every trait, there's a branch that goes to an organism. So for example, on this chart, before you get to any organisms, the first trait on the main line is vertebrae. And then the first branch comes off to sharks. Then you keep going on the main line, you come to another trait mark. It says bony skeleton. And then there's a branch that goes out to ray-finned fish. Keep following that main line, you've hit 4 limbs, and then a branch to amphibians. Amphibians have 4 limbs. Amphibians also have a bony skeleton and vertebrae, so they will contain all of those traits that were before them. The next trait on the main line is amniotic egg, and then it branches. And on the branch, there's the list of a trait for hair. So when you see this, where a branch is coming off and then only that branch has a trait listed, that means only the animals or organisms on that branch have that trait because it's only that branch where that trait is listed. So this branch actually forks at the end of it into primates and rabbits. So primates and rabbits, because they're on this branch that says hair, only those organisms have hair in the entire cladogram because only their clade or their branch says hair. So if we go back to the main line, we keep going. The next trait is eggs with shells. And then at the end of this branch, it splits into crocodiles and birds. And both of those organisms are going to have eggs with shells. So we'll look at some of the questions. Remember, plesiomorphic, the primitive or ancestral character. This character would be present in all of the organisms on the cladogram. So what is the plesiomorphic character in this chart? It's going to be the first character or trait that you come to, which in this case would be vertebrae. Then it asks, well, what are the apomorphic characters? Well, these are derived characters, so that's going to be everything else. Bony skeleton, four limbs, amniotic egg, so on and so forth. Well, then it says, what characters do the ray-finned fish have? Well, you would just follow the chart, and whatever traits you come to before you get to ray-finned fish would be included in the traits that it has. So as you follow the chart, on the main line, you hit vertebrae, then bony skeleton, and then to the branch of ray-finned fish. So it's going to have vertebrae and a bony skeleton. What does the ray-finned fish not have? That would be anything past it, right? Four limbs, amniotic egg, etc. They come after the ray-finned fish branch, so it doesn't have any of those traits. Let's see, where are we at now? What characters do the primates not have? Again, they're going to have everything but eggs with shells because that comes after. Which organism is in the out group? Which organisms are in the in group? Well, the out group is going to be that most primitive organism, which in this case is going to be the sharks. And then the in group is everything else. The fish, amphibians, primates, etc. So anyway, that's the kind of questions you'll be asked on these charts, on these cladograms. And the way you read it is just usually left to right, bottom to top. Look at the traits on the bottom and then what organisms have those as they branch out. You might see something like this. You know, this is the same thing. It's just written differently. It's more flat. But you still just follow the lines. They're like water pipes. And every time the water passes a trait, that's like the water picks up that trait and carries it to those organisms. You could think of it that way. And this is just a little challenge that I found online to just kind of quiz you on cladograms. And there's all kinds of things like this. But if we look at this chart, it's got a character table on the bottom, and this is what we'll be talking about in one of our labs coming up. It's a character table that has a list of organisms and the traits that they have. It's just like a table. And then above that table, they have a cladogram created, but the organisms aren't placed. And you have to place the organisms based on what traits they have. Well, on the cladogram, it has a list of traits. Amniotic egg is first, red blood cells, no outer shell, all have legs and wings. So then you have to look at the character table and place those organisms where they go. Well, if we look, the mammals only have one trait on this list, and that's the amniotic egg. Well, on the cladogram, the first trait you come to is amniotic egg, and there's a branch with a blank. So we're going to put the mammals on that branch because all they have is an amniotic egg. The next organism comes after you run into amniotic egg and red blood cells. So we need to pick an organism that has amniotic eggs and a nucleated red blood cell. Well, it looks like that's going to be turtles. So we'll put turtles next. The next organism, it says it has no outer shell, and it also has the RBCs, the amniotic egg. So for that one, that's going to be our lizards and snakes. The next organism all have legs, and as well as these other three traits, and that's going to be our crocodiles. And then the last organism has wings, so that's obviously going to be our birds. And then that's how our cladogram would look from left to right. So the question number one is, well, what is the order of organisms in the cladogram from left to right? Well, it's going to be the one that says mammals and then turtles, lizards and snakes, crocodiles, and birds. That is correct. Select each characteristic that the common ancestor of the turtle clade would have possessed? Well, the common ancestor of the turtle clade would be down here where this branch meets the main line. And that would have nucleated red blood cells and an amniotic egg. So we'll select both of those. And that was correct. Why are crocodiles placed to the left of birds on the cladogram? Because they have scaly feet like birds, because they're closer related, because they have every characteristic except wings. Mainly C is our best choice here, because they can't be placed after wings, because they don't have wings, but they have everything else, so they need to be close. So that would be the best choice. So anyway, there's tons of little simulations like this that you can do that are pretty cool to just practice your cladogram understanding. And in class, we'll actually build these based on character tables, and we'll actually create character tables as well. So anyway, thank you for listening. Have a great day.